Hi everyone, my name is Richie and I'm one of your peer mentors, as well as a summer intern for the Smithsonian Office of the Chief Information Officer Data Science Lab, or the Smithsonian OCIO Data Science Lab.
Hi, everyone. I'm Zachary Wilson, and I'm also a summer intern for the OCIO Data Science Lab. And this is Richie and I's presentation, Quantitative Ecological Research through Machine Learning.
So many of you may recognize us as your peer mentors, where we teach you the fundamentals of data analysis in R, and that is one component of our internship. And the other component is working as interns for our principal investigator, Dr. Alex White. Dr. Alex White is a biodiversity research data scientist for the Smithsonian OCIO Data Science Lab, with expertise in biogeography, quantitative ecology, and community phylogenetics. Our work with him involves quantitative ecology, which is a field of study that applies mathematical and statistical methods to ecological research and the analysis of ecological data. One of these methods is machine learning, which can be used to gain insights about biological mechanisms that might influence population density. This summer, we're working on optimizing a machine learning model that uses island-spotted skunk images as data points so we could get a better understanding of their habitat and population. This data can then help inform conservation decisions. Furthermore, we hope to expand upon this model so it can identify more species with accuracy and utilize alternate data. This project was initiated as a joint effort between the National Park Service, the Nature Conservancy, UCSB OEP, and the Smithsonian. Additionally, several UCSB-Smithsonian scholars have been previously involved in data collection and creation of workflows for this project.
Core motivation for our efforts is to develop a workflow that facilitates research adhering to the FAIR guiding principles for scientific data management and stewardship. The FAIR guiding principles are a set of research guidelines that allows a project's findings to be easily utilized by other researchers and projects. By ensuring our data is findable, accessible, interoperable, and reusable, our data will continue to remain impactful beyond the scope of our initial project.
An important component of adhering to the FAIR guiding principles is to adopt a metadata standard. For our skunk research, we have chosen to follow a camera trap data scheme called CamTrapDP. Our camera trap images run through a workflow that renames our images in order for it to conform to CamTrapDP standards. CamTrapDP is a standard set by an international group of scientists interested in data science for ecology. Adhering by CamTrapDP is crucial because it allows our images to be used by different researchers using camera traps. Below is an example of what our image names will look like after they've been renamed to the proper CamTrapDP standard.
Richie and I's internship focuses on the use of data science and machine learning to achieve our research goals. As such, the majority of our methodology is carried out on lightweight and portable edge computing devices, such as the Raspberry Pi 400, so that our workflow may be utilized at remote research sites. On these devices, we have familiarized ourselves with bash scripting in order to assign and collect metadata from our image data. And we have also gained proficiency in Python so that we may utilize a TensorFlow-like machine learning model to identify the species captured in our camera trap images.
We use these edge computing devices to run machine learning models that can identify species from images. The creation and use of our models from start to finish goes as follows. First, we create the algorithm. Second, train it. Next, validate it, then apply it, and finally share it. This summer, Zach and I had the opportunity to run a live demo for the 2023 EDIS cohort, where we had them run their collected data through this model and present their findings through visualizations.
To summarize our progress to date, we have built upon the work of past interns to solidify our workflow that gathers camera trap images taken throughout Santa Cruz Island, converts all image data to fit a standardized metadata format, and uses a machine learning model to identify species found in images with the goal of identifying sightings of the Santa Cruz Island spotted skunk. Moving forward, we are modifying this workflow to become modular so that a variety of machine learning models may be slotted into our existing workflow. We are working to make our workflow not just viable for image identification, but also for audio identification as well. Ultimately, our final goal of our internship is to have a general purpose workflow created that may be used for multi-format identification in quantitative ecological research.
Additionally, we plan to use segmentation for our machine learning model to assist with counting unique island-spotted skunks. We hope to use segmentation to generate mass, which are images that can help us look more deeply at the island-spotted skunk's fur. Individual skunks differ by the spots on their backs, so using segmentation can use this characteristic to help us get an estimate of individual organisms. We're optimistic that our work will contribute to significant improvements to this model, and thank you for listening to our presentation.